अब हम बात करेंगे reagents and supplies जिनकी हमें ज़रूरत पेश आएगी for transgenic mouse preparation. सबसे पहले स्पेशल इक्विपमेंट इस पूरे प्रोसीजर में हमें काफ़ी ज़्यादा चाहिए. एक तो हमें इन्वर्टेड माइक्रोस्कोप की ज़रूरत पेश आएगी which is different from the usual microscope. Usual microscope में जो lens है that is from on the top of the specimen or sample under study. जो inverted microscope है the lens is at the bottom or the objective is at the bottom. So the 40x and the 10x eyepiece is required. 40x objective is required and 10x eyepiece is required to perform this method, because when we have to observe the embryo or the fertilized egg, then this kind of magnification is the minimum requirement to proceed further. Then we need micro manipulator. There are two single instrument holders are also required, because when we are going to proceed further with this micro injection to the fertilized eggs, so we need these two joysticks which will control the movement of on one side injection, and on the other side we can operate that particular pipette which is going to immobilize the embryos during all this process. Dissecting microscopes are required, with regular dissecting microscope or one with eliminating from the base, because when we have to isolate eggs from the animal, we may need these dissecting microscopes. Then compressed air source, microforge, surgical instruments, controlled environmental chambers. These are the preliminary requirements before we can proceed further to start this method. As you know that the cells cannot be left unattended or dried, because once the water content is lost, then the cell cannot no more survive. So जो एम्ब्रियो है इसको जब हमने प्रोसेस करना है, वी डेफिनेटली नीड सम बफर्स, सम मीडियम which can keep these embryos or these fertilized eggs in the moist condition, and we can proceed with them. For that purpose, we will be using Whiten's medium, which is a combination of different salts like sodium chloride, potassium dihydrogen phosphate, magnesium sulfate, hydrated magnesium sulfate, then sodium bicarbonate, glucose, calcium lactate, sodium lactate, sodium pyruvate, BSA is the source of albumin, the protein, source of protein, sodium EDTA, HEPAS, and then the antibiotic penicillin and streptomycin, so as no infection or no contamination should be there. Once this medium is prepared, we have to aerate it in the presence of 5% oxygen, 5% carbon dioxide and 90% nitrogen, so as we can use it for further processing to keep the fertilized egg alive. And the shelf life of this aerated buffer is about a week. If stored on the shelf in a tightly capped container, then we can use it, and this storage should be in the refrigerator. Once the shelf life, that is 1 week, is passed, this buffer can no more be used. Besides it, we need balanced salt solution, sterile. Hanks solution is one of the example, and then we need DNA injection buffer that contains 1 millimolar trace chloride, 0.1 millimolar EDTA, and the pH 7.2.